Thank you everyone for coming to our first AI clinic for clinicians, specifically for people in Cambridge this time. We're very happy to welcome you here to tell you about AI, machine learning, and how it can help healthcare, and also tell you about the work we're doing at CCAIM, the Cambridge Center for AI in Medicine in Cambridge, and introduce you to a few of our tools that you might be already able to be using. And in the end, also answer all the questions you might have on the topic. This is our timetable for today. We will start with an introduction by the clinic and the center by Prof Andres Floto, one of our directors, and then go to talk about machine learning in healthcare, starting with Professor Mihaela, our other director of the center, who will talk about what machine learning can already do in healthcare and what the opportunities are for clinicians in the usage of that. Then we have Dr. Alexander Gimson, who will talk about machine learning in transplantation medicine. Then we have Dr. Eoin McKinney, who will talk about how you can utilize machine learning now, introducing a very exciting tool that we have just released to the world a couple of weeks ago, AutoPrognosis 2.0. So you can find risk scores very quickly for whatever field you're working in. So this is quite exciting. Then we have Evgeny Saveliev, who will talk about time series software and show some demonstrators, as well as Robert Davis, who will also talk about demonstrators and machine learning interpretability software. Afterwards, we have an extensive Q&A where you can just ask your questions. So at the end of it, you can either raise your hand, or if you have a question already during the talk that you don't want to forget, put down in the chat and we will read it out to our speakers in the end for you. Now I'm handing over to Andres, and I hope you will have a good clinic today.
Great. So on behalf of Mihaela, myself, the co-director of the center, a warm welcome to you all. So this is the first of hopefully many AI clinics where we hope to engage specifically with clinicians in the Cambridge area to begin with. I'd like to spend the next couple of minutes just describing the Cambridge Center for AI in Medicine, which many of you will have heard of, but is actually a very recent endeavor. We only launched at the beginning of last year, thanks to support from AstraZeneca and GSK. So multidisciplinary across the Cambridge biocampus, and really our mission, if you like, is to develop pioneering AI and machine learning technologies which will transform biomedical science, medicine, and specifically healthcare. And really we're looking at focusing on this sweet spot between medicine, machine learning, and science. That intersection really is where we believe we could offer more or most at this interdisciplinary cutting edge research. And we're passionate that it's really the challenges and problems that are present in medicine that drive the innovation in new methods in machine learning and AI. And conversely, we believe that the machine learning and AI can drive revolutions in medicine, and some of those are beginning to be realized. So just to spend a couple of minutes just introducing the team, and I think you'll get a flavor of how cross-disciplinary this is. So Mihaela is at applied maths, but also public health and medicine. Stefan from Judge Business School, Sarah Teichmann from Sanger, Owen, many of you will know from medicine, Miguel from engineering, myself from medicine, Pietro from computing, Ari from medicine, Angela from public health and HDR UK, and Alex from medicine. And together with this faculty, we've also got an associate faculty, including Namshik from the Milner, George from medicine, Xu Yang from pure maths and mathematical statistics, Adrian from engineering, and Richard from the University of Liverpool, previously at Roche. So within this kind of large faculty, we've got a huge range of expertise, both in terms of domain expertise, but also machine learning and at that cutting edge intersection. So our ways of working are different. So we want to break the silos that exist, this kind of problem of communication between data scientists on the one side and clinicians on the other side and deliver truly interdisciplinary research. We're really eager and keen to learn from and teach each other. And again, this is a kind of ask to people in the audience. If you have difficult, challenging problems that are not really addressed by existing methods, come and chat to us. Really, that's exactly what we're looking for, is ways of stimulating methods development within machine learning. We want to understand, formalize, and solve fundamental problems in medicine and healthcare. And as we mentioned before, the medicine itself will drive cutting-edge research in machine learning. We want to be path-breaking, fast-moving, and impact-driven. And we believe that within the center, we have a critical mass of faculty, and also importantly, a critical mass of brilliant PhD students, which we've been recruiting from the very best around the world. We hope that they are multilingual in the sense that they have expertise in machine learning, but also appreciate the problems of medicine and healthcare. And really, our research activity is focused on 4 pillars. So biomedical discovery, so basic biology, structural modeling, systems analysis, cheminformatics. We have a second strand around omics analysis, let's say how to integrate these large data sets that arise from genomic, transcriptomic, and proteomic experiments. The third is around population studies. Can we develop novel methods of clinical trial development, provide personalized therapy and prognostication for individual patients, methods to understand factors that could lead to early diagnosis or indeed other important clinical outcomes. And finally, developing methods to improve healthcare delivery, home monitoring, assisted triage, and developing learning healthcare systems. So that's the broad range of activities. Today, you'll hear a few examples of the work that we've been doing, and hopefully that should inspire you to think about these big problems, come to us with these, and hopefully be able to utilize the tools that the center is developing, which are easily available to anyone who wants to use them. So in the interest of time, I'll stop there and hand over to Mihaela for the main bit of the session.
Thank you very much, Andres, and thank you very much, everyone, for joining us today. It is really exciting to present to you today some of the machine learning tools that we have been developing together with clinicians and others in the center. Today, I'm going to go through a very rapid pace to show to you the landscape. Machine learning can be empowered, of course, by a variety of healthcare data coming from a variety of domains, whether it's electronic health records or registries. And we really envision machine learning to provide tools that can empower clinicians, but also patients, enable discoveries of all sorts, both pharmaceutical but also clinical discoveries. But we also see machine learning helping hospital administrators to enable better operations, as well as healthcare bodies or policymakers. So you should see machine learning as a tool that can solve problems in a variety of areas in medicine. I'd like to start by saying clearly machine learning can do medicine, and it is aimed at building tools that can provide trustworthy recommendations to clinicians. So it is aimed at empowerment, not replacement. And indeed, in my own lab, we have developed a series of engagement sessions with clinicians that we call Revolutionizing Healthcare, where we go beyond the clinicians in the center to try to understand complex problems in medicine, and then try to go back and try to develop these methods with machine learning. What type of machine learning tools we have in mind? They go well beyond what you may have heard in the media. So we do not just look at, for instance, imaging technologies and try to develop methods to empower radiologists. We also use machine learning to do dirty tasks, such as, for instance, dealing with complexities, errors, and biases in the data through data-centric machine learning. Methods that enable us to exchange data sets without privacy fears, such as synthetic data. A variety of analytics, whether it is building risk scores at scale, whether it's time series forecasting, whether it's causal deep learning, whether it's treatment effect inference, whether it's dealing with transferring something that has been learned in one particular ecosystem, one particular clinical domain, or one particular geographical area, and transfer it to another area. Also, we built, as you are going to hear more from Rob today, a variety of technologies to transform these tools from black boxes into white boxes that the clinicians can engage with, can debug, and can take into consideration when they make decisions. So the focus is to create a human-machine partnerships where machine learning can empower clinicians and more generally the entire healthcare ecosystem to make better decisions and function better. So I would like to emphasize the fact that we do not see machine learning just doing what we already do with statistics at times and better, but rather as a tool that can solve a wide range of problems, and some of them are operational in nature. If it is to start from the area of building risk scores and prognostic scores, what we have tried to do, since machine learning is increasingly used to build risk scores for a variety of settings, and people are starting to go beyond just regressions, we saw that there are many diseases, many variables, many needs, and hence what we decided to develop is machine learning technologies that can do the crafting of machine learning models for a particular problem and for a particular setting. And in this context, the solution that we arrived at is to build automated and integrated and interpretable machine learning methods. Together with Eoin McKinney and our lab, we have developed a tool that Eoin will tell you more about later, which is called AutoPrognosis version 2.0. And the idea here is that we can build prognostic scores at scale for any problem that you may be interested in. This is a follow-up of an earlier tool that we have developed a few years back, which was really aimed at crafting risk scores. And this particular tool was by now used in a variety of settings, not only by us, but also by others around the world. And here are some of the areas that we have taken this tool at. The initial ideas were from cardiovascular disease and cystic fibrosis with collaborators in Cambridge, and cystic fibrosis in particular with Andres. Then we have applied this to many diseases, and also inclusively to operational issues. As a matter of fact, we have been the first use of machine learning, I think, around the world, where we partnered with NHS Digital early on in the pandemic in April 2020, and you have used the precursor of the current AutoPrognosis tool do management of resources in hospitals. AutoPrognosis 2.0 goes a lot further, and you will hear from Eoin about that. But it's not only prognostic scores and using cross-sectional data. As Eoin often says, this is about time series data in medicine. So hence, what we really try to do is build within CCAIM tools that go beyond dealing with cross-sectional data, looking at time series data, as well as estimating clusters, temporal clusters, doing screening and monitoring, determining when we should be screening a particular person and potentially with what particular technology. And finally, technologies for early diagnosis, which are enabling us to identify early symptoms that may be associated with disease. Just to give you an idea of the type of things we are doing, we are integrating a variety of data, whether it's from electronic health records or from registries, and then we are able to identify how often we should be monitoring a person, what is the trajectory of disease of this particular person, and what are the competing risks associated with this. For this, we have built a variety of tools. Some of them you see here. They have been deployed on cancer with Vincent, on cystic fibrosis with Andres, on Alzheimer's disease, and also on hospital and ICU care with Ari. To give you an idea of the type of tools we build, we are using machine learning, deep learning methods that are integrating a variety of time series data and that are able to predict time to event analysis, not only for one event of interest, but rather to many competing risks. And this is important for elderly, it is important in complex settings such as hospital care and many other settings. And indeed, in recent times, we took this tool together with Vincent to estimation of progression during active surveillance for prostate cancer. This is one of the many applications of this type of technology, and you can read more about that if you would like. Also, the problem of time series forecasting is essential in complex settings in the hospital, where it has been shown that delayed ICU admission is correlated with mortality, and identifying late patients that are at risk and transferring them late is increased with ICU death. So what can machine learning accomplish in this context? It can identify earlier than the clinicians, when patients should be admitted to the ICU. This also goes the other way. It can identify early when the patients can be safely sent to the regular wards. And indeed, this is research that we have been doing for many years, even prior to the current establishment of the center, up to 7 years ago. And indeed, what you see here is that we can identify earlier than clinicians, when patients are deteriorating. If you are interested to see the variety of tools we are building, you can take a look at our research pillars, and also Evgeny will tell more about that. And also together with Ari Ercole, we have developed a package called Clairvoyance that deals with this time series forecasts and also identifying monitoring and how often we should be monitoring patients. But we do not only deal with forecasts and predictions, we also deal with interventions. So trying to identify from observational data counterfactuals, which will tell us what would happen if we would treat the person with one particular treatment or intervention or another, or what would happen if we treat later and we wait. Answering such questions from observational data is tricky because of both missing counterfactuals and confounding. And as a matter of fact, this goes beyond predictions. It's about causal effect inference, where again, the center and more specifically our lab has been a pioneering lab in the world in this area of building causal effect inference methods. And I'd like to highlight here, and Alex will tell you more about that, we are solving in this way complex counterfactual problems of the type what would have happened if we would have not treated a particular person, for instance, in the case of Alex, transplanted the person with this particular organ, but rather we would have waited for a different organ. What's the impact on the patient? What's the impact on the other patients? And also we can use this to do longitudinal estimation of treatment effects, trying to understand how to treat, when to give treatment, and also importantly, when to stop treatment. This can be done for a variety of diseases, and one of the ones that we have looked at is cancer. Another one that we have been looking at is combining pharmacological models with disease progression model for treating COVID. This is especially important because in COVID, repurposing dexamethasone for COVID-19 seemed to be very appropriate, but very early on, people identified that precision dosage is key. And we have used this type of technology to identify when and how and to whom we should give dexamethasone and in what dose. With that being said, I'd like to finish by saying that we have these engagement sessions with clinicians called Revolutionizing Healthcare. And the next one will be on November 21st at 4 p.m. And we often have CCAIM clinicians being the lead in these discussions. The next one will be with Eoin, and it will be on AutoPrognosis. Thank you.
Hi, I'm Alex Gimson. I'm a transplant hepatologist, and I'm just going to tell you in a few minutes some of the work that we have been undertaking with ML and transplantation. And I'll try and sort of point back to some of the points that both Andres and Mihaela have made, and also point forward to what Eoin's going to be discussing in the future. So the reason why transplantation is such an interesting area to think about for ML is there are a number of reasons. First of all, there are extremely good databases, which are both national in this country and international, which we can interrogate and use as our core when trying to develop new ML models in transplantation. These databases are highly dimensioned. There are an enormous number of both donor and recipient factors over long periods of time, which impact simple outcomes such as survival, graft survival, or patient survival. Many of those factors have non-linear interactions, which are obviously very amenable to ML methodologies. But most importantly, as Mihaela's just mentioned, we have to deal in transplantation with counterfactuals. If a patient has presented to them a donor, the question obviously is, what is their outcome with that donor at this point in time? What might be the outcome if they waited in the future for a better donor? And what might be the outcome if that donor was actually given to another of the many different patients that are currently on a waiting list at any one time? All those require quite careful implications about causal inference, how we can predict the future of a particular patient at a particular point in their whole life time series. So machine learning for transplantation is about problem solving some of the really difficult issues which we have tussled with over many, many years, where standard statistical methodologies may not have been fully successful or indeed accurate at predicting prognosis. So we have recently, with Jerome Berevitz and Felix Imrie and a number of others, developed some methodologies which we are now published at ICML and NeurIPS, which describe how you can do that with different techniques, and clearly were shown to be better than our standard statistical methods using Cox regression. And by, for instance, allocating organs using machine learning, we are able to show that total population life years, the total life years of a population on a waiting list, can be maximized over and above our current allocation systems. One of the exciting things also is, of course, that there are enormously important new areas which we can introduce machine learning to. And Mihaela touched upon that. And I think, as Eoin said, all medicine is time series data, because patients have histories and because patients have futures. And so using time series data, for instance, to interrogate different phenotypes of donor and recipient pairs is an incredibly important way of identifying, for instance, what levels of immunosuppression we should be using. It's most unlikely that we would need to be using exactly the same amount of immunosuppression across all patients. What levels of monitoring. Transplantation requires lifetime monitoring, which is enormously resource intensive, and better ways of stratifying that. So we monitor those who need it more and those who need it less differently. So the use of time series data is going to be really, really important. My last point, which I think is going to be discussed by Evgeny and colleagues in a minute, and that is that using these technologies, we have been able to develop demonstrators, so we can actually demonstrate to people exactly what the impact and the counterfactual impacts of all these different parameters of donor and recipient details might have on our outcomes, population life years, or survival following a transplant. So, in the interest of time, I'll leave it at that, and I'm happy to take any questions, which I think we're all going to have at the end of these sessions. Thanks.
I'm assuming that everyone who's dialed in here is dialed in because they've got a vested interest in AI or what it might be able to do for them or for their practice or for their patients. And fundamentally, there's been a huge problem here, and that problem is analogous to the drug development problem that was faced about 10, 20 years ago, which was described as crossing the valley of death and has been rebranded in the context of AI as the AI chasm. And that is going from the potential of what AI might be able to do for our clinical practice and actually getting across that chasm to the impact. And for many people, actually, they are on the impact side. So for much of the audience of who we're talking to today in this AI clinic, you're probably clinicians without an extensive background in software development or AI application. So you're staring back from the impact perspective, trying to see how you can use that potential to change your practice. And that's really what I want to talk about in the next couple of minutes. The question you may be asking yourself, and hopefully are asking yourself, is how can you use AI, but not just how can you use it, how can you use it properly and robustly? And there are increasingly regulations or guidelines that are pointing out to us how AI should be used. So for example, here you can see, and this has just come out in Nature Medicine. In fact, Ari in the center is a co-author on these recommendations. You can see that it's recommended that we don't just use ML or AI models because they're the newest, hottest thing off the block, but that we make retrospective backwards comparisons to simpler, conventional statistical models as well, that we're very clear about publishing protocols, very clear about publishing model performance, transparent models where the hyperparameters that are used and selected to tune a model, and there are many of those underneath the bonnet of an AI or ML algorithm, that those are very clear and transparent so that something can be reproduced and ultimately validated. And these won't come as any surprise, but they have only comparatively recently been formulated and formalized into sets of recommendations and guidelines for how AI or ML models should be developed for application in a clinical context. So that's all very helpful. And indeed, they've been more broadly defined in the context of applying AI for social good, and AI for social good is in fact a movement in its own right. And they stress things that you wouldn't necessarily inherently expect to see on the page there for AI applications. Right up at the top is simplicity. In actual fact, we shouldn't be striving for complexity as a goal. Simple solutions are often the best solutions, and the simplest solution that is most effective can be the right one. But there are a range of different possible tools that you might be able to select for a given problem. So there won't be one way to predict to develop a predictive model for the clinical problem that you're facing. There may be multiple ways to do that. There may be multiple possible selections of different tuning parameters that you may want to make in your data. There may be multiple different ways in which your data could be pre-processed before you even start to build a model. And all of that needs to be transparently handled and done up front. And that's something that requires a bit of experience around AI. So coming back to how should I get involved with AI? How should I use AI? Or used to be, and that's the point of what I'm saying here, there used to be a bit of a buy-in in terms of required knowledge and experience of algorithm development. Probably the major development that we're describing here is that arguably is no longer necessary. You can get involved with applying AI to clinical problems without the necessity to do that background work and to understand and tune and optimize. That can be done for you. And that's the automated pipelines that Mihaela has been describing. And that's why we think that is such a major step change in the accessibility of AI approaches for tackling clinical problems. This can now be done in a way that is no less robust than in fact, it's more robust than it ever would have been done before, but it can now be done by non-expert people like clinicians who bring that domain knowledge of the clinical area rather than the detailed background expertise of the AI algorithms themselves. So how can that be done? Because these guidelines are now 10 a penny. There are many different guidelines describing what we are striving for, what we should be doing. There's almost nothing that tells us how we should be doing this or gives us practical guidance on how to implement it. And that's what's changing. I was just giving the apocryphal examples of how things can be done badly, very high profile examples that are very successful at predicting breast cancer. But in fact, this led to a complete firestorm because shortly after it was published, it was pointed out that it was entirely non-reproducible because they hadn't developed their models transparently. It was impossible for anyone to reproduce it. And even the groups doing the best sort of work in this area have historically not performed to the sort of standards that guidelines are now setting for us. But it's now feasible for even non-experts to do things in the proper way, transparently, reproducibly, to build models that are interpretable, that can be debugged, that can be understood, and that can be accurate and can potentially change practice. So how can that now be done? Well, that's where this new tool, this automated ML framework can really help. It basically does the heavy lifting so that you don't have to. So the data pre-processing, the missing imputation, the scaling, the data preparation steps, those aren't just done by one single method. Those are done by multiple parallel methods with cross comparison between them to optimize it. And under the bonnet of any ML algorithm, there are, as I mentioned, many different hyperparameters that require tuning and optimization. And in fact, algorithms can be quite fragile. They can be quite sensitive to those input tuning parameters. But this automated framework optimizes selection of those hyperparameters so that you don't need to worry about it, so that you can point this automated pipeline at the data or the question at hand and let it do that for you. So it's been implemented, I think, fantastically as an open source, very easy to use, state-of-the-art approach. We're actually considering trying to test drive this with some school kids rather than necessarily with clinicians, because it is bulletproof in terms of how easy to use. So it makes these sorts of approaches accessible, I think, to the people who are probably interested in the sort of presentations that we're making today, to people with the domain and clinical expertise rather than the ML expertise. So we're not saying, bring data, we can do this for you. We're saying, here's a tool, go and do it yourselves, basically. So what it does is it takes the raw medical data, creates a curated data set, does missing value imputation, optimizes all of those upstream hyperparameters, trains a model, doesn't just train a model with your favorite, most up-to-date, hottest ML algorithm, but it makes direct backwards comparison to simpler models like Cox proportion hazards models, for example, simpler statistical models where they may be equally effective, they may be superior, they may be preferable to use in practice and easier to implement, but you get that side-by-side comparison, which is critical and missing in most other approaches. Fundamentally as well, these approaches are interpretable. In other words, it's not black box. It won't tell you that it's making a prediction and let you puzzle out why and just have to trust it that it's making the appropriate prediction. It will read out and tell you why it's making that prediction and how it reached that prediction, which is critically important because it allows you to then debug it and make sure there's no information leak and you haven't fed it information it shouldn't really be privy to that are influencing its predictive accuracy. But it also increases trust in the likelihood of adoption. And we know that if people understand why they're responding to a particular piece of clinical information, they're much more likely to continue doing it. And finally, it doesn't stop there. It goes on and builds a clinical demonstrator that allows you to interact with, play with, and visualize the predictions in a way that lets you unpick what's contributed to each prediction and to understand them in much more detail. And that makes them much more implementable as well. So I just move on. I'm probably taking too long. So what we think is that this automated ML framework now handles all of the key requirements, all of the key challenges that are traditionally put in the way by those guidelines that are entirely appropriate. There are stringent guidelines for how ML models should be built in a healthcare context, but they represent challenges that have to be overcome. Well, this automated pipeline handles each of those and makes building these sorts of models accessible to people who aren't experts in it. So each of these different areas are handled, I think, very elegantly. And the bottom line is at the bottom there. It's try it. What we want you to do is to have a go, really, and let us know how you get on. In order to facilitate that, there are regular sessions, the Revolutionizing Healthcare sessions, where these are unpicked in much more detail. The first that relates to the automated framework is already available online through the link that you see there. The second is coming up on the 21st of November, which is a more practical hands-on approach. However, the link at the top there for the AutoPrognosis website includes a detailed tutorial that will run you through exactly how you can and should apply this to data that you already have. So please use the links, have a go, and get stuck in.
I'm Evgeny Saveliev. I'm a research engineer at Cambridge Centre for AI and Medicine, and I'll briefly present an update today on our software and demonstrators that we have in the space of time series data. So why are time series so important? I think this idea has been mentioned a few times already today in different people's talks, but to quote Professor Eoin McKinney, just presented earlier, all problems in medicine should be considered time series problems. All of our patients have histories, all diseases have natural histories, and clinical experience tries to capture these trajectories, but almost no formal methods have so far been able to do so. And indeed, that is why exploring time series methods is so fundamental in the domain of medicine. And so on that topic, we are getting ready to release a new time series tool from the center, which is called Temporal, spelled with AI on the end, and we're planning to release this in January 2023, so do stay tuned for that. There will be updates, and this tool will provide an overall pipeline that includes time series data pre-processing, prediction and forecasting, time to event analysis and risk scoring, individualized treatment effects and causal inference, and evaluation and benchmarking of all these settings. In addition, it will also incorporate temporal clustering, so it's clustering of, say, patients over time, and interpretability methods, such as feature importance methods, which, of course, as was discussed by various people, interpretability is now critically important when it comes to machine learning methods. And the tool is powered by a wide range of methods that we have developed, and as well as a number of other benchmark methods as well. We are looking to incorporate data sets like the MIMIC ICU dataset and also the cystic fibrosis data, as mentioned earlier. We have ongoing research with Professor Andres Floto around this. Now, finally, we also already have provided a number of demonstrations of the time series problems and methods, which are linked here on the screen, but you can also find this information on our website. And two demonstrators that I'll highlight here, which are in the time series domain, is on the left here. You can see the individualized treatment effects demonstration, and on the right is a time to event analysis with temporal clustering demonstration. As well, the one with individualized treatment effects in the video, you can view it there on YouTube. It uses the MIMIC dataset as an example, and the one on the right for time to event analysis and temporal clustering uses a prostate cancer dataset as the underlying dataset. So hopefully these demonstrators are helpful for getting an idea of how a time series toolkit is applicable in medicine in practice. And that is all from me, quite short. And yes, thank you for listening.
So, hello everyone, my name is Rob Davis. I'm a research engineer at the Centre, and I'm here to talk to you just very briefly today about ML interpretability, something that we've mentioned a few times in the session today. Okay, there are some incredible ML methods that have been developed both here at the Centre and more broadly in the ML community, and they can do amazing things. However, these methods are limited, have a limited impact without interpretability methods running alongside them, particularly in a high-stakes environment such as medicine, where you need to have utmost confidence in a given prediction. Yeah, a black box model just isn't good enough anymore because it is such a high-stakes environment. There are also many different types of model. We talked a lot about time series, but the traditional ML interpretability methods are just simple post hoc explanations of static predictions. Again, that isn't enough anymore. We need multiple different methods to sort of span the full range of our different ML methods. So we need interpretability methods for time series, for patient trajectories. We need unsupervised learning methods for patient phenotyping. We need to interpret heterogeneous effects estimations for personalized therapeutics, and we need concept-based explainers to discover new clinical concepts. At CCAIM, we're developing a suite of different interpretability methods. The images on the screen are from our interpretability GitHub, that aims to gather together all of our methods alongside some key methods from the community and explain when you should use each method, help you understand them and implement them. As I say, we've got many different methods and many different interpretability methods for those, so we need to cover. These are all examples of tabular data explainers. We also have time series and unsupervised methods, and recently a new interpretability method for individualized treatment effects. Each interpretability method works in a different way to provide a different type of explanation that broadly breaks down into feature-based explanations, which are those that indicate the input features that were important to that individual prediction. We also have case-based explanations, that is methods that show you examples that the model sees as being similar to the input record, so you can compare and contrast that way. And we also have concept-based explainers, so that this is any method where, well, these are methods where you can provide concepts to the model and then see how important that concept was to the model's prediction. And so, yeah, by going through those, I hope you can see we have methods that are both generic, that they work across the board, and also personalizable, and by which I mean personalizable to the user, not just to the patient. So yeah, that's things like the concept-based explainer there, but there are others as well where you're providing your own context and then you see back an explanation in those terms. And so, yeah, this is just a quick talk, and I'd like to end on three things. The first is, if you follow the first link here, that will take you to one of my favorite methods from the suite, which is Simplex. And there you can see it used in action in a demonstrator app, just like the screenshots that Evgeny showed you. There's another demonstrator we have. So yeah, I'd encourage you to follow that link if you're interested. Second is the interpretability suite on GitHub. So those are the images I showed you, but there's a full breakdown of all of our methods with lots of information if you want to read about the different methods that we have. And finally, I'd like to say, just watch this space. There's lots happening in this area, and I hope to speak to you at a future clinic with much more detail on all of these different methods, and hopefully show you some more user interfaces like the demonstrator linked here.
All of our speakers as well. So if you would just like to join me on video, and then we go straight into the Q&A. And I will kick it off with a question that I just got by one of our participants, who's asking, whether you could share your thoughts on further qualifications or education beyond med school, that would be very helpful in order to enter the field of ML in medicine.
I guess the first point to make is, hopefully, with the advent of the automated frameworks, the need for those additional qualifications has declined substantially. In other words, you don't need to have the same expertise, and that allows you to make the most of the clinical domain knowledge that you have and apply the tools more simply. However, depending on your background, coding skills are going to be something that is useful. There are courses within the university, and in fact from the center we had our inaugural summer school just a couple of months ago, and that will be an ongoing feature. So there will be a summer school that you'll be able to participate, which will give you a thorough and excellent grounding in both the basics all the way up to the cutting edge techniques as well. So yes, that whilst that's not a formal qualification as such, you get a certificate for having done it, and more than that, you get exposure to both the basics and some pretty cool new stuff.
Terrific, thank you.
I have a very brief comment to add to that from more like a practical perspective, because when I started my move towards machine learning, it was very much kind of the thing that I did. So to actually become able to use these kinds of tools without going in too deep into theory, there's plenty of resources available online. In particular, I would recommend one called Data Camp, and then also I would recommend courses on either Coursera or Udemy. So those are worth having a look at. Very practical, but they really help you get into grips with the actual coding side of things.
Thank you very much. The next question by Timoleon. What type of visualizations do you use, and how did you choose these methods? I think it's in relation to the tools we giving out to people.
I guess I will let an Andres answer to this, but the idea was that these tools are very much general. So you hopefully can use them for a variety of diseases and a variety of settings, as I was trying to describe at the very beginning, maybe not sufficiently eloquently. These tools can often be applied for many different types of diseases and to answer a variety of problems. So I think it's more about identifying the class of problems you are trying to solve, and given a particular disease, we can tell you what data set one tool may be most appropriate. I don't know, Andres, and whether you want to say anything about the generality of these tools.
I think you've captured it perfectly, Mihaela. I'm not quite sure that there was a question around visualization of the data, meaning the outputs from the demonstrators. I'm not, or the
I think that was the question. Yeah,
is that the question?
I can briefly comment if that's all right. Typically in the space, specifically for the kind of things that we want to present, and this will include things like heat maps or various line charts or scatter plots, etc., as well as interactivity aspects of it, which is most relevant to the kind of tools that we release. There are really two big sort of tools that people use, and we use as well. It's either one called Streamlit or one called Plotly Dash. So have a look at them if you're interested in visualization stuff for machine learning and data science broadly. Those are good tools, good libraries.
Perfect, thank you. There's one question by Will about how our AutoML pipelines work with different dataset formats. You've mentioned flexibility, automation, and pre-processing. Does the model also handle the structural format in general, mimic, OMOP, CDM, custom registries?
So again, maybe I will jump in. So again, AutoPrognosis version 2.0 is aimed at cross-sectional data. Of course, you can take temporal time series data such as mimic and use it to make predictions for the next time interval. However, if you are interested in time series data, I would advise waiting a little bit for the tools from Evgeny to be released. We have a precursor of these tools called Clairvoyance, but we are upscaling to these new tools called Temporai. But the type of formats you can use for AutoPrognosis are actually very diverse, and you can even interconnect to AutoPrognosis through R and tools like that. I don't know, Eoin, whether you want to add anything else, or Timing, who is on the call and has used the tool as well.
Yeah, was the question oriented towards the structure of the data in terms of whether it's, for example, tabular text data or continuous covariates, or that type of data structure, or what type of data structure and format were you asking about?
Yeah, if you want to clarify, Will, feel free to unmute yourself and add to that.
Yeah, sure. So firstly, thank you. These have been really good talks, really obviously amazing work. And thank you for putting on all these sessions. I go to some of the other ones as well. They're really good. Yeah, the question was just around, I know, having like spoken to people doing ML research, and particularly on the medical side of things before, oftentimes just pulling out the data in the first place, like knowing what fields to extract, everything like this is a problem in itself. And so, yeah, so again, like Mimic versus OMOP or anything like this, I just wondered, I guess my question was more general, like, is there any plan down the line to be trying to tackle those issues as well? Or is that something that you see as being an upstream problem to solve, like data standardization, before it gets to your pipeline, or something that's handled in your pipeline, if that makes sense?
Yeah, it does make sense, and it is a challenge. And feature selection upstream is often guided by one or two approaches. You can do dimensionality reduction, you can do feature selection based on variable features across your data set that are most likely to be informative, or you can do domain knowledge selected fields that you know are likely to be of relevance. So there are different ways in which you can scale it down to only include the most relevant features. Not for different methods, feature reduction is not always a requirement. And there are variable, depending how many features you've got, you may have to reduce, you may not.
But I think, may I jump a little bit? Sorry for that. So thank you very much, Will, for this fantastic question. Yes, as a matter of fact, AutoPrognosis, the second version of it, can do all that for you, in the sense that you can yourself decide how many features you want to put in. And as a matter of fact, if you go to the website, you will see a video by the postdoc that was leading this second version of AutoPrognosis, where he's showing how you can feed in AutoPrognosis more features, fewer features, so you can explore as a clinician what is the value of information, how much advantage you may get by adding more features or less features. And it allows you to look whether you should use, for instance, regressions or machine learning, and what's the best, let's say, performance you can get from the features you do get. So it allows you to build this exploratory environment where you can look at how many features, what's the value of these different features, and how these features interact to the model. And we have automated the pipeline of dimensionality reduction, so that's done for you in the process. Maybe we can take this particular question to one of our revolutionizing healthcare sessions on November the 21st, which Eoin has said, where we could talk more about issues like that. But the answer is data. Let's say if the question is about how much data you should put in and what's the value of the different data, that is something you can wonderfully explore with AutoPrognosis version 2.0.
Yeah, thank you very much. Yeah, again, to everyone, if you're really interested in this topic, please come on the 21st of November. Just sign up over the Van der Schaar Lab website, and then we will keep you up to date. Next question, probably building on this a little bit as well, is how large a database or a data set needs to be accessible or useful for AI?
That's a great question. And I would say this is exactly what AutoPrognosis allows you to do, as Eoin very nicely said, because regressions are part of this particular tool. You can select to use just regressions. You can select to use a machine learning model or a set of machine learning models. So that type of investigation of what is the added value of machine learning as opposed to regression models, you can do that in a very systematic and clean way and reproducible way using AutoPrognosis. So the answer is, it depends on your data, but go do this exploration in a very systematic way in this framework where you can select only the performance, to view only the performance of regression methods. You can select a particular machine learning model or all the machine learning models and regression models together and see what's the added value. And again, I'd like to highlight that this may depend also on the number of features you put in. So this entire exploration of the value of information and the value of machine learning can be done within this ecosystem. The tool was exactly built to enable this type of exploration.
I think it's fair to say, Mihaela, that's rarely done as well. People tend to go straight for the complicated model because it's the cool new thing, whereas actually something simple could do it just as well. And actually, if the goal is to get across that chasm and get something implemented, it's going to be so much easier to implement a simple model than a complex one. So it's definitely worth doing that side-by-side comparison.
Thank you. And there's a question that will probably also be discussed in the future more, is by Roy, who's asking whether you could explain in which ways domain knowledge is actually used or captured in building predictive models, and whether there has been success with using molecular data like gene expression data in making those predictive models.
So yes, there are different ways in which domain knowledge can be formally incorporated. My favorite example from this is from the DREAM Consortium. So it relates to genomic data, which is what you're alluding to, where they've spent the last decade doing formal cloud-based challenges, where they release a challenge. They say, this is a genomic data set. We want teams out there in the cloud to build a model that can do some task. They hold back some data so that they can assess performance, and then they rank the models on the basis of their performance. The reason that's a useful example is that they've been able to take a decade's worth of experience using genomic model building and say, what are the features of successful models? Domain knowledge in the team that is building it is one of the top features. So if you understand the problem, you're much more likely to build a successful model. You're not much more likely to be able to debug it. And that's arguably exactly the reason why we need to remove that gap between machine learners and clinicians to make this work, because you need domain knowledge on both sides to be working together. So domain knowledge really is successful when it's implemented in that way. There are other ways in which domain knowledge is with natural language processing. For example, people try to formally co-opt that into model building. I haven't seen clear examples of success with that route yet, but I think it's an admirable approach. Mihaela, I don't know if you may know of examples.
All I want to say is that the center is doing a lot of work on the area of AI for more complex omics data sets of all sorts. So please keep an eye on the center if you are interested in that. And more generally, I think one thing that we are doing in the center is trying to bring domain knowledge. The example that we have been doing it recently in the center is bringing pharmacological models into machine learning as a form of prior knowledge, which enabled us to do machine learning on data sets with very limited amount of samples. So bringing your knowledge into these machine learning models in various ways is something very interesting to the center. So if you have innovative ideas on how you can bring your clinical knowledge, not only to debug models, but to feed these models, please reach out. We are keen to think about how humans, clinicians especially, and machine learning can interact to empower each other.
Thank you. We have like 2 questions left that we might be able to squeeze in. One is about the tool being used to decide whether a patient has to go to IT or not. And the question is this also possible for pediatric patients?
Yes, I do not see any reason why it's not possible for pediatric patients. As a matter of fact, Mimic does contain in one of the more recent versions quite a lot of pediatric patients. So I would say this is possible. This also alludes to this idea of transfer learning a little bit. how much can we learn from the adult population, which may be larger and better understood, and what can we transfer to the pediatric population, which may be less well understood. So these are interesting topics, very much at the core of research that's happening in the center.
Mm-hmm. And a question by Mark, in cases where the parameter is not identified, for example, undiscovered genotype, SNP, will AutoPrognosis be able to pick it up or suggest this could be an underlying reason if it wasn't fed in the first place?
So I'm not sure whether I understand the question, but if it is that you have a set of parameters and you feed them in inclusively a variety of SNPs, I think that this is something that AutoPrognosis could pick up, but be aware that at least AutoPrognosis so far was not tested with thousands of features, but rather more in the order of hundreds. This relates, I think, to other work that happens related to machine learning for omics data. I don't know, Andres, whether you want to say anything or
Yeah, if what you're asking is if you don't feed something into the model, can you identify what it is that you should have fed into the model? The answer is yes, by iteratively feeding it in. And it's a bit like what Mihaela said before, to understand the value of information and feature importance, you can do recursive feature elimination. So you can have a genotype, let's say, and your performance or your accuracy may be poor, and then you can add in a genotype and find that it improves. So but that's one way in which you can identify the value of the features you're including. Obviously, if you never put a feature into the model in the first place, you won't be able to evaluate its performance.
Let me jump to say one more thing related to this. So Rob talked about these concept-based explanations. So one way in which maybe you can identify things, so data needs to be there, but if you are referring to new concepts of disease, new concepts of staging of disease, new concepts of deterioration, that is possible post hoc, by interpreting, for instance, the predictions of black box model like AutoPrognosis with ideas such as concept-based explanations. So for instance, in recent times, for the sake of prostate cancer, we were able to show together with Vincent that one can recover some well-known concepts of prostate cancer related to staging, as well as some new ones. I don't know whether that, but of course the data needs to be put in. If you don't put the data in, miracles won't happen. But if the data is there, concepts could be uncovered, which may be unknown.
It also in some ways speaks to active sensing, Mihaela, in terms of identification of what features perhaps should be included. So that there are methods that can broach that if that's really what you're getting at. In other words, what's missing, what should be included. And Teaming has just put a comment in the chat there as well about another method being developed to identify which samples are potentially missing features, not which features might be missing across the entire set.
Perfect. Thank you very much. And thank you very much again to all our speakers. We close here. If you have any more questions, please feel free to reach out to us. And also, just to reiterate, we will have on the 21st of November our next Revolutionizing Healthcare session, explicitly on a hands-on approach with AutoPrognosis 2.0. So please come to the Van der Schaar Lab website, sign up for the session, and we will inform you via email with everything you need to know. Or come directly to our AutoPrognosis website, which is also in the chat, where you can read more about this. And come to the CCAIM website to just learn more about our general research, our other tools we're doing, and events that we're planning in the future. And I'm sure we will have further AI clinics like this to reach out to the clinicians of Cambridge and beyond directly. This video will also be available soon online, so you can share it with your colleagues and re-listen to some of the things you've heard today. And yeah, thank you very much for coming, everyone. And thank you again to our speakers. And I wish you all a very good day.